Last class we talked about, or really the past few class periods, we've been talking about what they call the central dogma of biology. Central dogma says you have DNA, and DNA is organized into genes. Those genes have the information to make proteins. Now, for efficiency's sake and for the sake of protecting the DNA, we make a copy of the DNA called RNA. And that process of making a copy of the RNA, that's transcription. And for eukaryotic cells, that happens in the nucleus. Okay? And then in the cytoplasm, we will, and using the ribosome, we will do translation, where translation is where we make the protein. Okay? Now, if you remember back to when we were going over the parts of the cell, I mentioned how we had ribosomes in the cytoplasm and ribosomes that were attached to the rough ER. The rough part of the ER, talking about the ribosomes that were attached to the ER. You may know the difference between proteins made in the cytoplasm versus on the rough ER? Luke?
Usually proteins made in the rough ER are sent outside the cell.
Yeah.
Whereas things made in the cytoplasm are sourced.
Exactly. And so the question is, well, how do we know which proteins need to stay in the cytoplasm, which ones need to go to the rough ER, and then, or are being sent outside the cell? And that's kind of what we're talking about here. Now, another kind of little, not as important detail, but it's this idea that we can do some slight modifications to these polypeptide chains. Once we make that final protein, there are some small little chemical groups we can attach to it that will play a further role in targeting it. So like in other words, like say you make a protein that needs to stay in the cytoplasm, we can add something to that protein that says where specifically in the cytoplasm does it need to go. Okay. This first bullet point is kind of talking about this idea that when you make that polypeptide is just sort of, you know, they call it beads on a string. Like it just looks like, here's the amino acid, there's an amino acid, right? And then that thing folds up into that three-dimensional polypeptide. And then you can then combine multiple of those polypeptides, and that makes the fully formed protein. Thank you. Yeah, so again, for that protein to be like a fully functional protein, there are some, they call them post-translational modifications. You don't need to know the details of them, but just kind of be aware that it exists. We'll attach, it could be a sugar, a lipid, a phosphate group, that will help it become that fully functional protein. And then we can even cleave or cut a polypeptide into smaller polypeptides. But not, kind of a little more of a asterisk, maybe not asterisk, but not like quite the full focus of like this part of the section, but just kind of more of a little random detail. But this is really what I want to focus on is how do we know like a protein that needs to be like sent out of a cell? Well, how do we get there? So here's what happens. Most every single protein, whether it's going to be sent out of the cell or it's going to stay in the cell, will actually begin translation in the cytoplasm, which is kind of backwards because what I made it sound like is you have ribosomes permanently attached to the rough ER, and those ribosomes there, they will make proteins that get sent out of the cell. But even if the protein is sent out of the cell, and eventually that ribosome gets attached to the rough ER, it begins translation in the cytoplasm. And during translation, there will be a set of like codons that code for amino acids that form what we call a signal polypeptide. So this signal polypeptide, it'll signal where the protein goes. So where protein goes. Okay? And when you have that peptide, there is a particle called a signal recognition particle. Also called SRP. So it's a molecule that'll recognize that signal peptide, and if this signal peptide says, hey, this protein needs to be sent out of the cell, or maybe this protein needs to stay within the endomembrane system. So this process, it's proteins that are going two places. They're either going out of the cell, or they're part of the endomembrane system. Remember the endomembrane system? That was like the where you had the ER and the Golgi apparatus. And it was like, all right, once you make a protein or something that needs to be sent out of the cell, how do we, ER, like it'll transport to the cell, we'll do some modifications. Golgi will then package it up, say where it needs to go, and then we send it out of the cell. So proteins that need to be part of that system. All right, so those types of proteins, you have that signal peptide, a signal recognition particle binds to that peptide, and if that signal peptide says, hey, this protein is part of one of these two systems, bless you, that signal recognition protein brings it to what's called a translocation complex. So a translocation complex, it is a protein complex, so multiple proteins in the ER membrane, and that signal recognition protein binds to that complex, and then translation, so translation's pause here. So pause translation, but then as soon as like that signal recognition particle binds to the translocation complex, translation proceeds again. We finish translation, I think is the best way to say it. We finish translation, and we're then, as we're finishing translation, we're now finishing translation, but we're sending the protein inside the lumen of the ER. So you finish translation, and the protein goes to the ER lumen. The lumen is like the inside part of the ER. Okay? And then you have that fully formed protein. And then this protein can go through the endomembrane system, right? Finish going to the ER, go to the Golgi apparatus, and then from the Golgi apparatus you would have vesicles that take it to the cell membrane, and then it goes out of the cell. Okay, this would be the system again for if it's a protein that's going out of the cell, or if it's a protein that's staying within the endomembrane system. Maybe it's a protein that functions in the endoplasmic reticulum, or it's a protein that functions in the Golgi apparatus. Okay, if it's a protein that stays in the cytoplasm, you would not have a signal peptide. So protein in cytoplasm. There is no signal peptide there. There's no signal peptide. There's no signal recognition particle to bind that signal peptide. And so this whole picture here doesn't happen. Okay? Now, I have a question for you. What if it's a protein that needs to be used in the mitochondria? How would that work? If you remember, mitochondria have their own DNA and their own ribosomes.
I just said that.
Yeah, good. So if it's a protein that needs to be involved in the mitochondria actually makes its own proteins. Okay. Questions about this? I think it's kind of a cool process. This signal peptide does get removed. I didn't mention that. So it's worth pointing out this signal peptide doesn't actually have a role in the fully formed protein. Its only job is just to tell that to tell the cell, hey, we need to send this protein inside the endomembrane system. This is really no different than just like a tracking label. Like it's just kind of a tracking thing to get this protein to go inside the ER lumen. Okay. Next few slides are just kind of saying everything in words I was just saying. Again, polypeptide, it always begins in the cytosol. Okay? So even if that protein eventually goes inside the endoplasmic reticulum to be sent out of the cell, it will always begin, that ribosome begins in the cytoplasm. Okay? A little bit more of a complicated picture than what you've been told so far in your biology classes. Cool. Shifting gears a little bit, and I've mentioned this before, that so we're doing transcription, right? We make our RNA, our messenger RNA, and then we'll do translation, make our proteins. Well, let's say I need to make a whole lot of protein. It would not be very efficient if I had to do like, okay, one at a time, one ribosome makes a protein, that ribosome then goes, uses that mRNA, makes another protein. It would be more efficient if like you could have multiple workers on the assembly line. multiple ribosomes making polypeptides at a time. So what would happen is here's your start of the mRNA. Notice how the start is the 5 prime, the end's the 3 prime. You would have a promoter here. That promoter would be a region on that mRNA that tracks the ribosome. This ribosome would then get going, start making the polypeptide. As soon as it gets over here, you then have another ribosome bind, and then it'll start making the polypeptide. Once it gets past there, another ribosome binds, and so on. That way you can keep doing translation, making multiple polypeptides at a time. And that's what they're trying to show right here. Oh, and they call this a polyribosome. Polyribosome is just like the structure where you have an mRNA and multiple ribosomes doing translation at once. And this that polyribosome, that happens in bacteria and eukaryotes. And it might seem kind of obvious, but like this whole process here, this doesn't happen in bacteria. Why? Bacteria don't do this. This is not, if bacteria want to get a protein to be sent out the cell, they don't put it in the ER. Why? Luke? They don't have an ER, right? Okay, it seems kind of odd, but like keep that in mind. This is only for eukaryotic cells. All right. Oh, I'm sorry. I said promoter earlier. It's a start codon. Once you have that start codon, not promoter. Same kind of idea though. It's like it's almost like the idea of like the polyribosome. It's like if you ever been to like a water park, and they like send you down the big slide, and it's like you're on that big raft, and once you get like far enough down, they'll like send the next raft down and the next raft down. That's kind of what's happening with the polyribosome. It would not be very efficient if you're at the water park. They're like, all right, we gotta wait until he gets all the way down and I remember like going to like a water park when I was like your age, and there was like this like super like I guess kind of by the book kind of like person who like they had to be 10 seconds down, like, and it was like really like 10 seconds for like how am I gonna catch up to them? Anyways, it was not very efficient, but yeah, all right. Anyways, that's kind of the idea though. Okay, so this is only in bacteria what we see here. Now, how you can make this even more efficient, even more quicker, is let's say you do transcription, and here this is assuming you do transcription, you make the whole RNA, then you do translation. But what bacteria can do is they couple transcription and translation. So they don't even wait for the whole mRNA to be made before they start doing translation. Right? So here's the RNA polymerase. It's making a copy of the mRNA. But as soon as it's made, it's made a copy of the mRNA. Boom, a polyribosome attaches there, and it starts making the polypeptide. Now, why don't eukaryotic cells do it this way? This seems really efficient. Why are we not able to do translation and transcription? Like, why can't we couple those together like that? I'll give you a hint. It's like the same answer as what I asked you earlier with like why they don't do the ER transport thing. So transcription Thomas? Yeah, exactly. They do everything in the cytoplasm. For us, we do transcription in the nucleus. We modify that mRNA transcript before we do translation. So that's one reason we have to modify that mRNA transcript. The second reason is just literally just location wise. We do translation in the cytoplasm, but we do transcription in the nucleus. Bacteria, they do everything in the cytoplasm. Okay? Those are really important. Like the AP exam, they like asking questions that like bring together multiple concepts. So that'd be kind of if they're going to ask you about transcription translation, there's like a decent bet they could ask you something like that because it brings together a lot of different areas of like the class. Okay. All right. I think we're good here. So, eukaryotes, we have that the nuclear envelope. That's like the outer part of the nucleus, that double membrane. Okay, this picture here, this slide and the next slide, same picture, but I broke it up into two separate ones just so it could fit on the slide. It's just an overview of everything we talked about so far. Overview of protein synthesis. So, beginning with transcription, you have DNA. DNA is organized into genes. Each gene is going to code for a protein. So we then will do transcription, where we make that RNA transcript. Remember, we make that RNA transcript from 5' to 3'. And then, if this is a bacterial cell, they can just go straight to translation. Eukaryotic cells, they have to do RNA processing. RNA processing is where you remove the introns. So introns are removed. Intron, N stands for interspersed, so it's the parts that don't code for the protein. Exons stay. So exon stands for expression. It's the part of the mRNA that is expressed. Okay? And we then have the poly A tail. What end is the poly A tail at? 5' or 3'? 3'. Okay, good. 3'. And then 3'. And they don't show it. Well, they do at the very bottom here. You have the 5' cap. 5' cap. What is what's on the 5' cap? Guanine. Guanine. Yeah, 5G. All right, the 5' cap, the guanine cap. Those will help the cap and the tail. They help direct this RNA transcript out of the nucleus. Proteins will bind to it, transport out of the nucleus, and then you'll have so this is mRNA. You then have tRNA. So here's your tRNA. Remember, each tRNA is specific for an amino acid. We have 20 amino acids, so you'd have 20 different tRNAs. There is an enzyme called amino acyl tRNA RNA synthetase. That is the enzyme that will make sure with the proper amino acid gets attached to the proper tRNA. Okay. Once these then get made, they call these a charged tRNA when it has the amino acid attached to the appropriate tRNA. All right, so I'll go to the next picture. So continuation is just kind of shifted down now. So here's that charged tRNA. So right here, here's your charged tRNA. It's holding an amino acid. So we're out in the cytoplasm now. The ribosome is gonna the small subunit of the ribosome. Remember, there's two parts of the ribosome. The small subunit, that's gonna bind the mRNA, and it's gonna initiate transcription. What amino acid would this be? Luke? Methionine, yeah. How'd you know that? Yeah, we met in August, right? Methionine is always the first amino acid. What was the codon that went with methionine? When did we meet? August. AUG. Okay. All right. Then in the large subunit, so the large subunit, that's where you then have APE, the 3 sites. So that comes in. The A side is standing for the amino acyl sites, where the amino acids come in, right? The new amino acid in that chain, it comes in. The P site, so the A side is the next amino acid. So think A for amino acid, the next amino acid. P site, P for polypeptide. That is where, and you can kind of see this like the little tunnel that the polypeptide comes out of. P side is where the polypeptide is, the growing polypeptide is located. E, what's the E for? Exit. And that is the exit site. That's where the tRNA that's delivered its amino acid would leave. And then once this tRNA in the P side is done, it is then going to pass on that polypeptide to the tRNA in the A site, and then this tRNA that was holding the polypeptide, it then goes to the E site once it's transferred the polypeptide over, and it'll leave. Okay. What's the direction that the ribosome moves across the mRNA? 5 prime, 3 prime. 5 prime to 3 prime. What is the direction that we transcribe RNA? 5 prime to 3 prime. Same direction. It's like a trick question. I think that's everything. A little vocab. Remember, this is the anticodon. The anticodon recognizes the codon. The codon is on the mRNA, and it's every 3 bases, nucleotides, codes for an amino acid. I think that does it. Any questions about protein synthesis? Okay. Moving on to, I think, an easier topic, and that's just on mutations. I say easier just because it's really just, this is basically just like freshman bio stuff. But, you know, these are free points in the AP exam. So, remember, a mutation, that's when there's some sort of change in the genetic material in a cell. So some sort of change in the DNA. Now, a point mutation, that is when you just change a point. A point is just like one thing. If I do multiple points, that's called a line, right? It's not a line mutation, it's a point mutation. You just change one nucleotide. And nucleotide pair, right? You change one nucleotide, that automatically changes the other one, since they're complementary. Now, why that's important is if you just start, like, imagine a book. If you just start going and changing letters in a book, you change the meaning of that text in the book. Same with DNA. If you start changing nucleotides in the DNA, you're going to change the protein that DNA codes for. Okay. This is super important. This last bullet point. If you get a mutation like in your skin cell, like let's say you get skin cancer. It's really tragic if you get skin cancer, if you get mutations in your skin cells. But it's even more tragic if you were to get mutations in gametes. Why? Gametes, hey, these are the sex cells, sperm or egg. Why is it even more, why is it more bad if you get a mutation in a gamete? Trini?
Because then it can be like passed down to your kid or something.
Right, then that gets passed down. Okay, that mutation then gets locked in. So I want you to think of evolution here. Why do I want you to make that connection to evolution? Mutations in gametes, why do those fuel evolution? Mutations to skin leading to cancer, it's really sad. I don't like that, but that doesn't lead to evolution. Mutations in gametes, that will lead to evolution. You got to make that connection, but why is there a connection there? Did you? Go ahead.
If one of your gametes gets like passed down, there's no like recessed or dominant trait that will like pass it.
You're thinking very, very. I'm just like even more general than that. Why would mutations in gametes fuel evolution, whereas mutations in your somatic cells, the opposite of a gamete would be a somatic cell. Those are your body cells. So literally, like mutations to think of there's like the idea of like a weightlifter, like lifts really big weights. Say a big bodybuilder has a kid. You never go to the gym. You have a kid. Whose baby's stronger? You would think the body, but it's not. No, neither person's kid is stronger. They're all the kids got to go to the gym by themselves. Okay, that's related to this. Tom's good.
But the somatic cells only affect you, but the gender sex cells will affect your
Exactly. Yeah, really, really latch on to this. These get passed on to the next generation, to the offspring. So if there's a mutation in a gene that's part of a gamete, that mutation can then, think of like the fruit fly experiment. That mutation can then get passed on to the next generation. But if it's a mutation that's not in an actual gamete that that can get passed on to the next generation, it might impact things in terms of like the number of offspring you have. Like say you get a mutation in your body that like somehow helps you reproduce more, which is very, very rare in like one like lifetime. That could lead to evolution, but most mutations, if it's not occurring in the sperm or egg, something that can be passed down, it's not something that's going to impact evolution. All right, I'll keep it moving, but really important idea there. All right, we've actually talked about this a little bit, so I'll try to go through, but we talked about sickle cell, I think twice now. This will be the third time, but sickle cell is like an abnormally shaped, yeah, Trini?
I have a question about like the whole cancer thing. So since like skin cancer, so if you're like breast cancer or something, you get skin cancer and it's like it doesn't affect the gametes, does that still like? Because they say if your family has a history of it, then you have a probability of getting it. Is that just because like you have like maybe they got it easily, so then you can get it easily because you have like similar DNA structures, even though it doesn't really affect like the gametes, or is it like something?
Basically, yeah. Like if the reason why, like say the reason I get skin cancer is because I went and tanned every day of my life and like fried my skin cells with a bunch of UV radiation. That's why I got skin cancer. That has nothing to do with my offspring. That's not why they would then get skin cancer. But some people, they might be at an increased risk genetically of getting skin cancer because maybe like the actual mechanisms in like their cells, like the genes that would code for proteins that like fix DNA damage, if there's like something locked in there where they don't make maybe enough of, or again the proteins that are supposed to change mutations, because if we make a mutation, we have enzymes that can correct these. So say like where the genetic part of cancer can come in is like that part where like you maybe your genetics, maybe you have better genes that like your body is really, really good at fixing and recognizing mutations. Or maybe your body is really, really good at not making mistakes in the first place. Your polymerases are even more accurate than somebody else's polymerases. That's how like kind of the genetics of cancer can kind of come in. So that's a really good question. All right, anyway, so here we're seeing a point mutation. So if we change, it's just a single letter. This should be a T. If I change this to an A, remember we code for amino acids with triplets, with codons. So now this would then code for GUG on the mRNA, which would code for the amino acid valine, VAL, and not glutamic acid that you would need for the normal hemoglobin. So appreciate just how subtle this can be. Changing one amino acid drastically changes the shape of an entire red blood cell and the fate of an entire organism. That's how like dangerous one single mutation can be. Not all mutations are like that. Like a lot of mutations, you change a random letter, it doesn't actually change. It might change maybe how often we use a gene, but it may not like break it to this degree. Okay. A little vocabulary for you. What is this strand here? This top strand. What do we call that? The blank strand. Template strand. Okay. What does that make this the other DNA strand? The coding strand. Notice how the coding strand is the same as the mRNA. Well, unless this was supposed to be, unless like here, this is a T and mRNA be a U. Okay. All right. Moving on. So when we're talking about mutations, there's two broad categories they fall within. There is a single nucleotide substitution. You swap one, you swap an A with a T, or there can be an actual insertion or deletion. You will add in DNA or you remove DNA. Okay? This one right here, I'm going to star this. This is the most dangerous. When you insert or you delete a nucleotide pair, that is the most dangerous. And I'll explain why it is, but make sure you make note of that. Insertions and deletions are the most dangerous kinds of mutations. All right, so let's look at these with like just 3 examples. So here would be what we call our wild type. Wild type is, when we look at the fruit flies, that means there's like no mutations with it. So I got my DNA strand, my template strand here. Here's my coding strand. Here's my normal mRNA and the protein. Amino end, carboxyl end. Which end is the amino end? 5' or 3'? Thomas? 5', good. So that means carboxyl end is the 3'. Remember carb, that's where we add to the polypeptide. We add to that polypeptide at the carboxyl end. Anyways, here, this left side, we're looking at if we just do substitutions. Right side is looking at insertion or deletion. There's like maybe 3 pictures, examples of these. So here, substitution, this is kind of, well, sort of like what we saw with the sickle cell example. So here, instead of this being a G, we made it an A. So because of that, that made the coding strand be a T. And so now, mRNA, you don't have a T, you have a U. And this is what they call, this example right here, they call this a silent mutation. So it's silent mutation. It's a type of substitution, but the effect is called silent. Here's why it's silent, because now we code for a stop codon by putting a C there. So it was CUA. AUG, UUU, GGU. This now codes for a stop codon. Oh, wait, I'm sorry. The reason why they call it silent is because we changed this DNA sequence. So here, we made this now be GGU. Up here, it was GGC. Okay? By changing that third amino acid from C to U, it didn't actually change the polypeptide. That's why they call it a silent mutation. Think of it this way. Like, I really hope that all students get their work done. But like, if I'm going to have a student not get their work done, I'd rather they be the silent type. Right? They don't disrupt the entire other class. They're just silently not doing their stuff. Right? Silent mutation, you technically change the DNA, but it doesn't actually affect the actual protein. Okay? The actual phenotype of the organism, what the organism looks like, they still look the same because you still make a functional protein. Silent mutation. All right. Now looking over here, this is going to be an insertion. So here we add an extra A nucleotide. When we do that, we now cause a frame shift. So this is why I said insertions or deletions are the most deadly, is because they cause a frame shift. Because think about it. Up here, we went AUG. Then you went AAG, UUU. If I were to add or remove a letter, I now change which 3 I read. So now everything is messed up from here on out. Because now by adding in that extra A, this now codes for a U, and so now everything gets shifted over, and now I hit a stop codon, and now we're done with the making that polypeptide. They would call this nonsense. Nonsense is when you code, when you change the DNA, either you insert or delete a base, or you substitute a base, and it codes for a stop codon. That is a nonsense mutation. It's nonsense because this is nonsense. The whole thing just stopped. You didn't even make a protein. Okay? Let me keep moving on. I'll show you the other ones. Another kind of substitution they call a missense substitution. So this right here is a missense substitution. So what happens in missense is we change one base, but now we code for a different amino acid. Okay? So now this codes for serine instead of a glycine. So now, this is what's tricky with missense. Missense, sometimes it can be a very, very drastic change to that protein. Missense is what you saw in sickle cell anemia, where you change one amino acid, and sometimes that could completely change the function of the protein. Other times, it just sort of just changes it. It's not like a big deal one way or another. Now over here, this would be another example of missense. So missense applies to both of them. Missense and insertion and deletion is we remove one of our, this is a deletion now. We delete an amino acid, or not amino acid, a DNA base. Now we have missense because now you're making a bunch of different amino acids. But this is frame shift missense. So understand why it's frame shift. By removing an amino acid, we're shifting where we're reading this. Now the whole thing is messed up. By removing a letter, you have now changed all, like what is now a grouping of 3 has everything has changed because you removed one single amino nucleotide. Okay. All right. Last kind would be the looking at nonsense with substitution. So missense, nonsense, silent. Those can apply to substitution or insertion or deletion. Just want to be clear with that. So this is nonsense with substitution. So here, we change one letter, and now it's nonsense because by changing that one letter, it hits a stop codon, and we're done there. Okay? Here, now this is the 3 nucleotide. So there are 3 nucleotide insertion or deletion. This is the least harmful. This is the least harmful kind of frame shift. Why is it the least harmful? Well, I said least harmful frame shift. It's not even a frame shift. Why would it not be a frame shift? What did it do?
You're making like a whole codon, so like 3 of them are back together, so it's not really just one.
Exactly. So it's not harmful because there's no frame shift. You added or you removed an entire codon. So all it would do was, if you look at your normal sequence here, you had lysine. All we did was you just removed lysine. But everything after that is the same. So this would be deletion. Insertion would be the same principle. I could insert something, and now let's say I do an insertion here. All I would be doing would just be adding another amino acid, but everything after that would be the same. Okay. That's really about it. This is everything just in words. Last thing, it's just a little vocab. Mutagens, that's any sort of agent that causes a mutation. Okay? If that mutagen causes cancer, that mutagen would be a carcinogen. Now, most mutagens are carcinogens, but they don't always have to be. Technically, not all things that cause mutations are also carcinogens that would also cause cancer. Okay? And we have a lot of, there's a lot of research methods that have been used to like, detect, like, all right, you know, a lot of like the FDA would look at this, like chemicals added to our foods, like trying to test to make sure, hey, does this chemical additive we put to our food to increase shelf life, is it a mutagen, and especially is it a carcinogen? Is it something that's going to be changing our DNA, which could increase the risk of cancer? So that's kind of part of what the FDA tries to do, is identifying like cancer-causing things in foods, or like trying to set limits on like, well, how much of this cancer-causing thing can you have in foods before it becomes like super, super risky for humans. Okay. Last thing, and we're not going to do the other section, by the way. We'll leave it here. But last thing here is just coming back to that definition of a gene. So they call it a discrete unit of inheritance. I said earlier a gene codes for a protein. You could think of it more broadly as a gene is a discrete unit of inheritance, right? You could think of it as a specific nucleotide sequence on a chromosome, like the list of A, T, C's, and G's. Or you could think of it, like I was saying earlier, DNA coding for a specific kind of polypeptide. Now, technically, genes can also code for RNAs, because we have genes that will code for the tRNAs and for the rRNA, what becomes our ribosome. That's why, like, technically you can't say a gene codes for a protein. Because you also got to think about there's other portions on a gene that don't actually code for a protein. It's kind of a technicality thing, but just something to keep in mind. that genes don't always code for proteins. They can code for some RNAs, and there are other portions of DNA that are like non-coding. They don't always code for a protein. Okay, I'm sorry. That went a lot longer than I was thinking, but the rest of the time, if you just do the mastering bio, we'll deal with 15.1 next class.